Well, it's great to be here in Budapest and to see such a strong community of people combining users and developers. And I'm old enough to remember when seeing something like Marvin programmed in Java was considered quite an innovation, and are using it in a pharma automation project. And so it's interesting to be here today and see some of the latest developments, which we'd like to consider how we might incorporate in what we've been doing. So my talk is basically trying to explain to you what we're doing in terms of integrated testing strategy developments in the area of alternative methods, where we're trying to evaluate the safety of compounds using a combination of in vitro and in silico evidence. So an integrated testing strategy is both in vitro and in silico, and it may be used for design purposes. It may also be used for a regulatory purpose. So that's why it's important we really think about how we put the evidence together. And as part of that, we've been looking at how we include good chemistry modeling and cheminformatics tools. So that's sort of the integration with some of the ChemAxon tools. So basically, this is part of a business innovation that we call ToxHQ. I consider it more of a consulting research organizational development rather than contracts, because it's very much a knowledge-based work. And we're located in Basel, in Switzerland, where we're looking at how we can put together good quality in vitro evidence with in silico for this safety assessment purpose. And the knowledge construct that has evolved in this field the last few years is called an adverse outcome pathway. It's a simplification of our knowledge describing how a series of events lead from a molecular initiating event. I show the one here for skin sensitization, where basically in an exposure situation, you put a product, say, on your skin. It could be your sun cream, it could be your shampoo, and that's your exposure. And the first molecular initiating event might be an interaction of that chemical with the protein in the skin, which then triggers subsequent downstream events, like you see here, activation of certain kinds of other cells, which may then lead to an immune response, a memory in the person's skin. So that kind of exposure in the future can lead to an allergic reaction, like a contact dermatitis. And so companies in this area obviously do not want that happening in their consumer product. They're no longer allowed, for example, in the European market, to use traditional animal testing. So they have to use new scientific methods. So the problem is, how do we combine in silico and in vitro evidence against these events to make a classification judgment on the safety of such an ingredient? And to do that, we're basically using a lot of analysis of biological data. We might, for example, mine transcriptomics data to get sets of gene signatures, to look at perturbations on network models. We might propose assays for particular key events. And so there's also a research dimension of developing this kind of ab initio-based risk assessment. And what you might do is you may have a key event in your propositional assay, and the in silico models may suggest that, or knowledge mining of the literature may suggest it, and then you would propose, okay, well, let's run a certain kind of bioassay to fill in the missing evidence. But what's the mathematical framework for weighting that and judging the value of information in the experiment you propose or in the model you want to use? So that's the overall framework. In many cases, the endpoint, like for example, an adverse reaction in liver from repeated exposure over a long period of time that might, say, lead to liver cancer, is a very complex framework and sequence of events, and we're still very much in the research area trying to figure out how to do that. The skin sensitization is relatively well understood, and that's the one that I'm talking about here. So one of the key problems is not just providing a judgment, but can we really understand and document the different sources of uncertainty in the result? Let's go back here. And one source is like the topic John was talking about at the start of this morning, is the lack of reliability in our integrating different kinds of data into support of an application. So that's something we've been working a lot on in the context of developing open standards with open talks as a community, and now building deployable containerization of different sources of heterogeneous data. And that may be part of your evidence against this kind of adverse outcome pathway. And then what you want to do in terms of supporting risk assessment is to integrate that data into a decision-making framework, like I'm showing on the left-hand side, where people designing a product think of both the potential hazard and the exposure, so they can design a safety level for the compound. So I want to show this this other slide as another point. So there are different sources of uncertainty in our data, not just our data science practices, but there's also variation because I'm showing as we vary for different replicates here, for different doses and time points, what kind of, after you've done the analysis, what kind of perturbations in the biological networks are we getting? And if you change the liver cell line, you get a very different response. You look at it at a different time, you also have a very different response. You run the same experiment in the same lab, especially if you're using cell lines like stem cell lines, you often get a different result. So some of the uncertainty is just fundamentally in the biology and its complexity. Now back to this specific example, one nice thing with skin sensitization, and we're partnering on this with Procter & Gamble, is they've worked very hard on understanding this adverse outcome pathway. There are validated assays for the key events. And with Joanna Jaworska, who we work with on this, they've developed a nice training set and Bayesian network approach for weighing the in silico and in vitro models. And what we wanted to do in this phase of the project, which we just released with some ChemAxon components at a show in the States in March, was could we basically create a reproducible application that could reproduce the work that had been done in the past, but more in an engineered application that could be extensible, and we could continue to improve as time goes on. And that's basically the current story in progress. And I think there's plenty of opportunities to continue to improve it. But as a baseline, we wanted to see if we could reproduce the work. And as part of that, by the way, Ahmed, unfortunately, couldn't be here today, is the lead developer on this with us. So if there are any really difficult technical questions, I'm going to get his help for you. But one of the things he judged was trying to include some of the calculators of properties like log P, log D, and log S from the ChemAxon calculators and see if he could integrate them into this Bayesian weighted evidence of in silico and in vitro data for the skin sensitization and see if he could reproduce the results. So that's basically, which I think we've been reasonably successful on, more successful than I've been on pressing this button. And this is the way the kind of network structure looks like. And you're also seeing with such a data set, what is the influence of a particular prediction from a model or the value of information in a new assay if I run it, because some contributions have a stronger effect on the end prediction result than others. So the idea in this kind of work is to quantify those weights, which are model development, and then also to advise the experimentalist there would be great value if you ran this assay on these compounds, because we'd really reduce the uncertainty in the prediction. So that's the testing strategy, which I summarize here. So not only do we want a weight of evidence with weights on the contributions from the in vitro and in silico components, but in the end we want to give a confidence with the prediction. So that's basically what the testing strategy is. And the goal is to produce this classification, basically 4 classes. The compound either does not sensitize weakly, moderately, or is strongly sensitizing. If it's strongly sensitizing, then the company probably decide we won't use this compound. However, if it was weak, they could use exposure as a design criteria and perhaps accept that. Obviously, non-sensitizing is favored. And what we show here then is we wanted to see if we could reproduce, with this workflow, the classification results and going through and replacing various components. So just showing that when we did that with the ChemAxon calculators for, I think it was log S, log P, and log D. This is the log P one. And then I have also, I'll leave the slides with you so you can take a look at it. The log D calculator replacement. And we had basically some minor differences, especially in a couple of more tricky intermediate classification of compounds. But overall, we were really quite pleased on being able to reproduce the accuracy in the published paper. And this gives you some kind of idea of the overall kinds of accuracy that we're getting. It's harder problem is the weak and moderate predictions, which I think we show in this summary table, that we do pretty well with the strong sensitizers, but there's still some challenges in distinguishing, say, between moderate and weak. And so in terms of these integrated testing strategies going forward, there's the goal of trying to improve on them, and there's also the goal of trying to get regulatory acceptance. And in the regulatory acceptance, we also need more of a convergence, and we need traits like clear reproducibility. And it's not just a predictive model, it needs to be considered as evidence. So this Bayesian network approach is part of that, trying to quantify this weighting in the evidence. And we've made a version available that you can try out if you're interested. If you go to that web address, that itsdouglasconnect.com one, and you can then draw a molecule or take a look at one of the molecules from the training set. It'll calculate the predictors. You can enter experimental data if you have it. And we're making that available as a web application so people can try it out. But we've also containerized it so it can be deployed to a local machine or intranet if you wanted to try it out within your organization. So it's not just a web application, in case you might think you couldn't use it locally. And basically the application does the calculations and then gives you the answer back of what your classification is. We also could potentially use it on other endpoints, other testing strategies, other training sets. So we're very interested in both on the partner side with continuing to improve the in silico calculations and also the in vitro evidence and how this can be used for different sorts of problems. So you can take a look at that and try it out. And we collected some statistics and just put it out there on the web with some messages around show in the States a few weeks ago. Got some very nice feedback on how it could be used by industry and adopted. We've since added deployment and reporting, so there should be a new version very shortly where you can also get detailed reporting on it. So in summary, I think the approach shows the kind of solution that is integrating, combining evidence in a way that can be used for this kind of application where we're needing to assess in a reliable manner the safety of a potential ingredient in a product. And that could be a cosmetic ingredient, but of course it could also be other kinds of molecules, like a drug-like molecule. From the web application we showed you, we also tried to present it to a kind of user who may not be a computational chemist. They may be more on the product team and thinking about a safety assessment. So they're not going to maybe understand all of the calculations. So we try to give, with web application, something that's user-friendly for those kinds of users to use. And if you're interested in trying it out, you basically have the possibility of looking at this on the web. We'd be happy to talk further in more detail about how this could go forward, and very much opening to partnering on it, because I mean it fundamentally needs, I think, partnering, because I mean no one organization has all these models and assays and so forth. So it's also needing to be integrated in that kind of way. And like I pointed out, if you wanted to try it at home, we can talk about giving you a locally deployable version at this stage. And sorry Ahmed isn't here to answer the hard questions. He's really the one doing a lot of the hard work on this. And there's our addresses if you want to email us and ask us some questions online.
Thank you for a very nice presentation. I think you got really reasonable accuracy given the relatively small size of your data set. I just had one question. When you showed the ITS results, you showed how you got much better accuracy for the strong kind of irritants compared to weak and medium. One reason could be that kind of this group of strong irritants may be less diverse compared to the other categories. Is this something that you checked out? Can you comment on that?
It's a nice data set, but it's still only, as you saw, we're on the order of magnitude of 100 molecules there, and that is an issue. You'd like to have thousands, but of course there's the issue of how would you get the data to validate it. But there are efforts going on in the cosmetics industry, realizing that different companies have somewhat different chemical space in their compounds. They also have a consortium called Cosmetics Europe, and they're trying to currently do some tests to evaluate the kind of issues like we're talking about by including more chemistry. And I think there's a strong motivation for them to collaborate on that. So that's one of the goals. We're trying to take a fairly open, I think, transparent approach with our contribution, is to help provide this convergence, because the overall industry goal will be to, I think, improve the strength of the training set and the method and the convergence of it, so it'll have greater regulatory acceptance and reduce that time frame. So absolutely. So there's still plenty of work to go on in terms of improving the applicability domain of the models, and even doing modeling I didn't get into in this area. There's plenty of room for further developments and improvements.
How difficult is it actually on the market to get regulatory approval? And do you think such modeling approaches to safety will be used possibly like or approved by regulatory authorities, or is it a bit of a stretch so far?
Oh, so it's obviously it's emerging area, and if you think of the three R's goals, that as a principle, it's to reduce, refine, and replace animal testing. So there is immediate business case in reduction, for example, and we also have a stronger scientific evidence, right? So there are immediate business cases. On the regulatory acceptance, we already have regulatory acceptance. So in this area, this kind of skin sensitization evidence on a chemical can be submitted under REACH Annex 11 guidance to a regulator, the European Chemical Agency in that point. It does have an OECD review as well. The issue there is every company still has some variation on their workflow. So 10 companies, 10 integrated testing strategies. So that's one of the issues I was mentioning. How could we have better consolidation of data and a better convergence on a consensus best practice? So that obviously is a hindrance to the increased regulatory acceptance. The other might be the increase in the data size of the data set. We do have Inform and that acceptance by the FDA of under ICHM7 such consensus in that case only in silico, right? It can be potentially accepted for that endpoint in terms of a drug impurity molecule and evaluating its mutagenicity. So we're starting to see this increased regulatory acceptance for certain endpoints, and that's going to be, I think, ongoing and a developing area in years to come. And some of the challenges I think the in silico community have to engage in is not just the issues on data we pointed out, but also on this topic of reproducibility. How can a regulator accept an in silico workflow if they can't reproduce it? And a lot of our data analysis today with our current methods is often not reproducible. So that's another challenge I think we as a community in silico-wise need to work on.